day, sir. Otherwise, shall we start with the second presentation?
Yeah, I think that's better. Definitely, technical thing can be sorted out.
Yes, yes.
By the time the introduction and Dr. Prakash presentation about Dr. Kurkure will join.
Yes,
and
yes.
we have almost 77 people are there, so we should
Right.
start. Yeah.
Dr. Swati, I think you can proceed.
Okay, sir. So very good morning to everyone, respected scientific community who have joined, and our guest speakers, and also the avian health professionals, and also the students and scholars who have present, and especially the executive committee of the AAHP. A warm welcome to you all to this Poultry Health Webinar series conducted by Association of Avian Health Professionals. Today will be the last webinar for this year, and through our last presentations or webinars, eminent speakers have shared their expertise and recent developments in regarding this poultry diseases, vaccination, managemental practices, etc. And today, our guest speakers are Dr. Nitin V. Kurkure, who's a Director of Research of MAFSU, who is a veterinary pathologist, and Dr. Prakash Baira Reddy, sir, who is the technical DGM of Veterinary Biologicals, who is a veterinary microbiologist. But before we start our webinar series, I would like to request Dr. D.K. Day, sir, who is the Vice President of AAHP, to give his opening remarks.
Yeah. Good morning, all. And I'm super excited to see that Hyderabad is the host of the next national seminar of AAHP. And let us take a pledge that all of us will make it a grand success in the poultry capital of India. Now, as we have eminent speakers. Yeah, I'm audible. Yeah, hopefully.
Yes, sir. Yes, sir.
Yeah. So I always say, maybe even at the cost of keep repeating the same thing, I always feel that in the poultry health, veterinarians are the most important professional team that takes care of poultry. As you might agree with me, even if the best nutrition and geneticist, with due respect to their efforts, can fail on the ground in one of the most dominant poultry market, that's India. I don't want to repeat about about statistics, but you should agree that with the depleting water resource and land resource and the poor water quality across, the stiff challenges faced by the poultry health veterinarians, and often they are not in prominence. I think Dr. Kataria and Dr. M.R. Reddy took this initiative of forming this association, which addressed the issues of this particular segment of veterinarians who contribute immensely. And I think today we are talking about One Health and AMR. But then that is true. And at the same time, if you see, we always say that we are working for poultry industry. But I feel maybe we are working for the food industry. Because whatever we are doing, our contributions are going to the food chain, whether it is egg production, milk, and broiler chicken or meat. Ultimately, the consumers are going to take those proteins. So there lies our importance, that how can we address AMR issues and One Health issues. So I think this webinar and in the future our national seminar will talk about the importance. And my only submission or request is that each one of us should work seriously and with more commitment about the extension, that talking to the farmers about the minimal use of antibiotic, and we talk to the human health fraternity and poultry health fraternity, so that we talk about One Health. So I'm excited to hear from Dr. Prakash Reddy as well as Dr. Kurkure. Over to you all. Thank you so much.
Thank you very much, sir.
Yeah.
Now I'd like to introduce Dr. Prakash Baira Reddy, sir. He is a veterinary microbiologist who have completed his Doctor of Philosophy, and he have over 20 plus years of experience in vaccine research institutes and biological industry. His research was most focused on control of emerging, re-emerging, and exotic diseases of poultry in India with a practical approach of lab-to-land programs. And he have a professional experience at different cadres from the Godrej Agrobate and in the Institute of Animal Health and Veterinary Biological Bangalore, and also in different cadres in veterinary biologicals of Venkateshwara Hatcheries Private Limited. And his research experience was surrounding this development of biofilm-based vaccines against colibacillosis, salmonellosis, and pasteurellosis, which are the burning problems of the poultry industry nowadays. And he have also secured an academic awards for his best research in MBSc and PhD in veterinary microbiology. Today, he is with us to speak about the infectious bronchitis virus variants and reviewing the global experience within Indian insight. I would like to request Dr. Prakash Baira Reddy, sir, to start with his seminar. Thank you, sir.
Thank you, madam, for the kind introduction. First of all, I thank the AAHP organizers, Dr. M.R. Reddy, Kataria Sahab, Day Sahab, all the organizing committee for inviting me to speak on the present burning problem on infectious bronchitis variants. So, am I audible, ma'am?
Yeah, yeah.
Yes, sir.
Okay, thank you. Okay, so I am working on this infectious bronchitis for more than 15 years to 20 years from now, which was the most neglected virus according to me in the initial stages. Though it is the number one disease after influenza, though it is the number one leading disease, if you take the mortalities of influenza, if you remove, because of culling, if you remove that, infectious bronchitis is the number one disease which is affecting the poultry industry globally. But in India, this is the most neglected virus according to my particular experience. So let us try to learn about the global experience and the Indian insight. I will try to give some inputs. So this particular virus has really gained more importance post-COVID. As everyone know, in 2020, when this particular COVID emerged in the global this thing, so the first industry which was affected was poultry. So based on that, this particular infectious bronchitis gained a lot of importance and people started knowing more about infectious bronchitis. That is what I interpret in this particular situation. Okay, so going ahead, let us try to learn about the infectious bronchitis variants, which is present globally and what is the Indian scenario. Okay. Okay, so before going that, where we are going around in this particular infectious bronchitis control programs, there is a lot of lessons we need to learn from the COVID outbreaks, which has happened in the human these things. So including when this particular COVID virus started spreading, how this particular virus was contained or the quarantine measures were taken, a lot of screening was happened and there were lockdowns. That is how we initially restricted the virus to particular places. So though it was restricted, it spread all over the world. Okay, because this particular virus had an incubation period of 14 days, so that it started spreading without knowingly. Okay, so that is how. So from the lessons learned, some of the things which we need to adapt in our poultry industry is first is the biosecurity, which everybody knows how the COVID was restricted. So same thing applies to the poultry industry, but it is the most neglected thing in the poultry industry, the biosecurity. Okay. So then comes how it was diagnosed. Diagnosis of disease is most important, but in human beings, when we are diagnosing COVID, we use a lot of the ultra-modern techniques like real-time PCR and then sequencing to know the variants which are emerging out. All those things need to be adapted in the poultry industry also to actually diagnose and contain this particular disease. Next, we need to identify the right serotypes, pathotypes, genotypes, which are different these things which we need to identify in a proper way and control accordingly. So there are certain things like vaccines which has been used. If you take the COVID vaccines, we have a lot of things to know. Like we can't use the SARS virus which emerged in 2002 or the MERS which emerged in 2012-13. So we can't use those viruses as a protector type for COVID. That everybody know now. So the such sort of things, lessons we need to learn in the poultry industry to control IB also. Okay, how the vaccine failures happen. All these things I will try to address in this particular seminar. And also in poultry industry, if you see, a lot of abuse of vaccines and vaccination schedules are happening. Okay, these things also we should learn from COVID. The simple thing is, even after the outbreak, whether we are really giving that vaccines as a prophylactic, we need to learn from the COVID stories. Okay, so in poultry industry, it is completely opposite. Even during the case of outbreaks, we are trying to use the vaccines and complicate the things. And last but not the least, the comorbidities and antibiotics. Whether during the COVID outbreaks, a lot of secondary bacterial complications which affected the lungs and the respiratory tract, which has happened. People are chronically affected with these particular comorbidities with other bacterial infections, which needs to be contained with the antibiotics in those cases. So these are all certain neglected things which we need to learn and adapt. Let us try to deal with some of the things in the So when we talk about the Coronaviridae family, okay, it is basically classified into 4 particular genus, that is alpha, beta, gamma, delta. The alpha and beta are more related to those viruses can cause problem in the human beings also along with the animals. But when we talk about the poultry, these particular viruses are more related to the gamma coronaviridae, that is gamma coronaviruses. Okay, so they have never affected the human beings in their particular things. Okay, if you talk about the alpha corona and the beta coronaviruses, what we know is SARS, which emerged in 2002. This is which has been highlighted as Sarbeco virus. Okay, this is the highlighted virus, that is the SARS, severe acute respiratory syndrome, but it vanished out within 6 months. The next emerged in 2013 was Marbeco virus, which is related to the alpha virus only, which is alpha coronavirus, which is called as Middle East respiratory syndrome, which caused in 2012. It also completely vanished. But of late, which emerged in 2019 in China and spread across the world, was the SARS coronavirus 2, that is COVID. So compared to these 2 viruses, that is SARS, which emerged in 2002, and MERS, which emerged in 2012-13, this particular virus is much milder compared to these 2. So this particular COVID virus was much milder compared to these viruses. That is why it has been spread globally, as the incubation period was also very low and the spread was, and this thing. That is why the reason. Low pathogenic viruses spread a lot. High pathogenic viruses, they are contained to certain place if it is controlled in a proper way. Okay. So why this particular things are happening? Let us try to know some of the characters of this particular virus. It belongs to a single-stranded RNA viruses. They are highly, changes can happen very frequently. Changes can mutate very fast. So each replication of this coronaviruses can lead to 3 mistakes. They can do 3 mistakes because their RNA polymerase capacity, proofreading capacity is very low. So that's why it leads to a lot of changes which happen in this particular virus. When we talk about infectious bronchitis virus, it is very highly labile virus and high mortality and high morbidity and low mortality. That is the character of this particular virus. But if it is complicated with mycoplasmas or other bacterial infections, the mortalities, secondary infection can lead to high mortalities. And also there are certain infectious bronchitis viruses, nephropathogenic viruses, or some virulent viruses. They can cause more problems also. There are some exceptions. So it is easily transmittable with a short incubation of 16 to 48 hours. So you can see how fast it can spread the virus. Okay. And high variability with high mutation rates. It is approximately 10 to the power of 4 substitutions per site per year. Okay, that is very important. I told you. So each replication can make 3 mistakes. You can imagine how many mutated viruses will be circulating around the world in the present context. And the second major thing with the variability is because of recombinations. We will try to talk more about this particular viruses. So when we talk about the IB pathotypes, we discussed about the character of virus. Now let us concentrate more on the IB pathotypes which has been emerged. The name infectious bronchitis virus has come from these particular lesions related to the respiratory tract. Especially you can see this is one of the postmortem lesion where you can see the plugs in the bronchi. These were the very typical lesions the people used to find with the infectious bronchitis virus, which was based accordingly, like infectious bronchitis virus. But over a period of time, as the time changed, this particular viruses started giving more other pathotypes also. Like these are all some of the published things at different timelines, which has been initially when it was identified. It was a classical virus related to the respiratory tract. Then it started affecting the kidneys in 1940s in Australia as a nephropathogenic strains. In 1950s as a heterotropic that is affecting the oviducts. And 1960s enterotropic strains emerged in different countries and the myopathies in 1990s. This is broadly identified pathotypes in different countries, okay, for the initial stage. In 2000, proverticulitis in some of the Chinese isolates, and 2010, the cystic oviducts, that is, which causes the water belly with the accumulation of fluids in the cysts, as well as ascites syndrome in the layers. This is some of the typical lesions. So in India, how exactly it has emerged out, let us try to see. After respiratory forms till 1998, we started getting a lot of reproductive forms, okay? With reproductive forms in the early age, if some of these viruses affects in the early age less than 4 weeks, they can lead to the fibrosis of the oviducts, leading to when it comes to the laying, you can find a lot of cystic oviducts, like false layer syndromes, you can say. So, which will have a mature ovaries, but it will be accumulated with the fluids because the ovum will not be able to enter into this thing. We can find a lot of peritonitis issues and impactions can happen. And even we have found a lot of cystic oviducts as early as 4 weeks also. You can see this is one of the postmortem lesions. You can see the cystic oviducts with nephritis in these cases. So that is one of the reproductive form what we have seen. The other form is in the later stage, in the growing stage, if it is affected, they can lead to a lot of immaturity cases. You can see this is a 28-week-old flock. A lot of immature birds are there with the respiratory infections. Okay, and you can see ovaries are not matured, and some of in these cases, if it is affecting in the laying stages, leads to a lot of misshapen eggs, and the quality of the eggs will be affected, especially the album, which becomes the watery album. So the other form of the infection is the nephropathogenic form, which has emerged in Australia in 1940s, but in India, we have seen this particular nephropathogenic strain emerging out after 1998. Okay, so this is particular typical signs we have seen in broilers, commercial layers, as well as the of nephritis cases leading to visceral gout. The China and the other countries with the proventriculitis and gizzard erosions, we have seen in India also such sort of pathotypes emerging. So other things is the myopathies. These are also the septicemic conditions we are finding such sort of cases also. This is the overall picture of the cases which is emerging in the IBM. So what is the effect of this particular IBM infection in the long-lived birds? And decrease the egg production, leading to delayed sexual maturity, drop in egg production, smaller eggs, egg peritonitis. All these cases people would have seen in this particular things, but majority of the time, even influenza. A lot of confusions do happen in the diagnosis of this. You need to think about this particular things also, leading to egg peritonitis, mortality, less hatchable eggs, all these things. And, birds which are having a sexual delay in sexual recovery is very slow, and sometimes it is irreversible damage. Eggshell quality, that is what I told, the internal quality of the eggs will be affected with watery albumin, misshapen eggs, and the shell quality, if you see, shellless eggs, cracks, deformed, all those things. If it is affecting in the breeders when we are going for the hatchability, definitely the low hatchability because of the loss of excessive loss of moisture and carbon dioxide in the shells, all those things. Sometimes people interpret as a vertically transmitted disease, though the viruses doesn't transmit from the parent to the chick. People interpret as a vertically transmitted diseases, but it is not like that. It will affect the quality of the eggs, leading to a lot of stunted chicks. Definitely, the chicks' performance will be very low, whether the broilers or when it comes to the performance will be very low in this thing when the parents are affected with the infectious varieties. This is how the things happen. So this is about some of the pathotypes which are varying, which we have seen. Now coming to the what is creating this particular changes in the pathotypes, which is creating problems. So this is because of the variants which are emerging. As I told, it is a single-stranded RNA virus. Okay, there is one particular gene called spike protein gene, S1G, in this particular, which codes for the spike protein on the surface of the virus. Okay, so there are three hypervariable regions in this particular spike protein gene, which are highly susceptible for variation, which lead to a lot of changes in the serotypes. Okay, so you can see a lot of changes in the genome happen, leading to the different serotypes. Okay, what about the serotypes related to this particular variations? So if there are more than 95% change in the spike protein gene, it is related mostly to the different serotype. That is how the things are being standardized over a period of time. So this is a standard, but that is not the same with all the serotypes or this thing. I will talk about those things in the next stages. So the best example to know the variants is the COVID virus. Okay, once it emerged in 2019 in Wuhan in China, based on that, it has led to emergence of a lot of different variants around the world, the alpha, beta, gamma, delta, so kappa, all these variants. And of late was the Omicron, which was the last which we identified, and we have seen that. So in India, we have seen the emergence of these two variants, kappa and delta, which has spread around, in the neighboring countries also. But we had a major damage with delta viruses of COVID. Okay. So this is some of the examples which we need to correlate and learn from these things. And see, this is how different countries have emerged with COVID viruses of different variants. So people think Omicron was the last globally which had emerged to be a variant which has spread a lot. But if you see, after COVID, we have seen more than 9 variants emerging out. Though it has been circulated, but it has not gained so much importance because of its low virulence. So the variant which is happening, it is a low virulent compared to the earlier things. Of late in 2003, you can see there are two different variants which are being circulated. That is EG5, nicknamed as Eris, which is dominant in US. The another new strain is BA286, that is nicknamed as Pirola. So this is what we need to know, how the variations happen in the COVID virus, which we need to now learn about the infectious bronchitis. So this is the recently published literature about the global prevalence of different serotypes around the world. If you see, the dots which are depicting or identifying some of the strains. Like here, the green dot, the mass variants, that is the GA1 lineage, we call it as, according to the recent classification. You can see this is spread across the world. So why this has spread across the world? Because we use a lot of live axes of mass. Though mass was first identified in 1930s, till now it is prevalent because a lot of variants are still emerging either from the vaccines or the mass variant. That is one of the reasons, including India. Okay, but if you see some of the countries like Australia and New Zealand, they have not introduced this particular strain in their country, and they have more of a local strains. We'll talk about that in the next day. The second most prevalent, which is spreading across the world, is the QX, which started in China in 1990s, and it has spread to the European countries and yellow dots in every country. They are the local variants which are prevalent in that particular country. Including India, we have our own local variants which is emerging and circulating and which is changing in that particular countries. You can see, including the US or Canada or South American regions, every country is affected with the local variants. So this was the classification earlier, which was classified into 32 lineages, okay, according to the changes which are happening in different countries, where a lot of local strains have been given the names of the lineages. Including India, we have our own lineage, which is called GI24 lineage. It was published in 2016, but I will show you the updated situation of the lineages next. So we have the GI24 lineage, which is circulating in India, apart from the US or Australia or any other country around the world. So what do you mean by lineages? It is very simple to explain about the lineages based on the human facial structures. You can see there are different types of people who are being bred and who are present in different countries. The changes which has happened based on the genomes and the environmental challenges. IB viruses. So you can see the lineages. That's what I was trying to show you. So
some problem with the audio?
Sir, internet problem, sir. He's trying to start his presentation now. Please be with us, please.
Sorry, I was disconnected. Yeah. Sir, now I'm audible?
Yes, sir.
Yes, yes.
Not able to open. So I have joined. I have. Sir, am I audible?
Yeah, you're audible, but you have to share your screen again, I think.
Yeah.
Was disconnected.
Okay now.
Okay now.
Yes, yes.
So now when hope the earlier slide it has been continued, right, sir?
Yes, sir, please.
Okay. So now when we talk about the lineages of of infectious bronchitis, if you take the North American, there are lots of Americans.
So sorry to interrupt you, sir. Your audio is still having problems, sir.
This is the European isolation. Now, am I audible? Hello.
Yes, sir. Now it is okay, sir. I think we are hearing you interruptedly, sir. Voice is not clear.
Yeah, those who have unmuted, please mute their voice. That may be one of the reason. Those who have unmuted, please. Those who have muted, please unmute the audio. Prakash Reddy garu.
Am I audible now, sir?
Yeah, yeah.
Yes, sir. Yes, please.
It's
Sir, can you switch off your video, sir, so that your net will be uninterrupted?
Okay.
Yeah. Thank you, sir.
Okay. So to continue, sorry for the interruption. When we talk about the lineages which are prevalent around the world, it is So this is the North American lineages which are prevalent in that particular region. Okay, the local variants, and this is the European variants. Okay, GI 28, 29, GI 21, which is more circulating in the European region. Okay, so this is the South American region. So you can see some of the variants in the Brazil that can cause infertility issues in the males also. Mexican variants, all those things are there. The recent one is the GA30, which is causing a lot of, without high mortality, they are causing a lot of production drops in the long-lived birds. Okay. Similar to what we are finding in India also. This is the Middle East variant, which is more circulating, GI23. Okay, this is the African variants, the Nigeria GI26. And you can see the Asian continent, you have a lot of variants, including the Indian one, that is the PDRC, which was identified in 1998. And now a lot of changes has happened over that particular strains, which are prevalent, which I will be talking in the next. And you can see the Malaysian, Thailand isolates, or Korean, Japan, they have their own local isolates which are circulating in that. Okay. So this is in Australia and New Zealand, which is circulating. If you see, every 20 years, they have a major variant which is emerging, which is more local only. There is no other global variants which are prevalent in the continent. So that's why I always say, think global and act local, what is prevalent in the country, which exactly I talk about the GA24 lineage, which is circulating in our country, which is very important. So this is the latest classification. You can see almost 40 plus lineages have been updated till now. Okay. So in that, if you see GA24, which is prevalent only in India, this is the Indian, this thing which is circulating in India. So we have mass variants, the 4/91, that is GA13 lineages, which is circulating. And there are some this thing about QX, but I will talk about that particular things. Okay, so this is the most prevalent, which is more than 80% of the isolates are related. So when we talk about this particular lineages which are circulating in India, this is the classification which is made according to taking all the standards into the consideration. We have three major genotypes circulating. One is the mass type, which is the mass variants. We have use of mass like live vaccines which are prevalent, which is this thing. The second is the Indian novel nephropathogenic types, that is the GA24 I talk about. And the third, which is more circulating, is the 4/9. Let us see what is happening with the Indian local strain, that is GA24 lineage, which was first identified in the 1998, that is by PDRC. So you can see these were the group of isolates which were earlier prevalent. Okay, but this has undergone a lot of changes and has left to the changes, further changes within that particular group, leading to this particular NPR variants, which we call now. According to the latest classification, if you see GA24, you can see a lot of isolates from PDRC or the Directorate of Poultry Research, and Dr. M. Reddy Saab has submitted, and a lot of other institutes like IVRI have submitted in this genotypes, or the Chennai College also. Third lineage which I was talking is the 4/91, that is belonging to the GA13 lineage. You can see a lot of isolates are there which are just without any changes they are seen. These are all some vaccinated frogs which has created an outbreak. So that is why there is a flat this thing which has happened in this particular group, the 4/91. Okay, so there is no changes compared to the vaccine strain. This is the homologous vaccine strain 4/91, which has led to the lot of the nephropathogenic. And there are certain isolates you can see here which are further classified within this particular group, and these are, we have identified these things are a recombinant of the mass and the 4/9. How exactly it happens, I will talk in the next slides. Okay, so talking about the GA24 lineage, which was published in 2016 by Mark Jacob, okay, and his group, you can see this has been classified as the indigenous to India, okay, part of which is from 98 to 2013 at that time, but now we have, I have shown you the further classification of this particular viruses. First publication was by Pyrie et al. In 2005, which was first time to identify this. So next is in the field, there is a lot of confusion that all the cystic oviduct cases, they are relating it to the QX type. Whether the QX is the only variant which causes cystic oviduct, I will try to explain you. So if you see, there are several variants. This was the first variant in China, which D388, which caused the cystic oviduct. So this was related to the QX type of strain which emerged and causing the cystic oviducts in the commercial days, the initial stage. But later, there are several isolates which has emerged in different countries like the IS-885 in Israel or the Delmarva strain in US or the North American region, where this has led to all the cystic oviduct type of cases in different countries. If you see, these are all the published different lineages causing cystic oviduct cases. Like the MAS variant, some of the VAS variants can also cause cystic oviducts, Australian T strains, the QX strains, the G823 lineage from Israel, Delmarva strain from US, and the G824 novel Indian NPR variants that has emerged out to cause this particular pathogen. So there should not be any confusion that the QX is being circulating, which is causing this particular pathotype. All these particular lineages can cause cystic oviducts. Let me be clear in this. And there are certain genotypes, genomes which have been submitted in the gene bank from the Indian claiming that to be a QX, but it is a very short sequence and it cannot be classified in the recent classification to be mapped in the genotypic lineages. You can see these short sequences are matching with different variants. Each virus which has been published in Indian you can see here, this is one of the strain which it is matching with the GI18 variant of Japan, JP1. And this is also the two isolates are grouped in JP1. The other one is grouped in GI13, that is the 4/19 variants, that is Italian 02. The other strain which has been published has been classified in GI27 variant, okay, which is more related to the Georgia 08 classification. But we cannot say that this particular strains are related to that unless and until we go for a complete sequence of the spike protein and try to map it where exactly is matching. So it is not a right time to interpret the QX, And this particular, whenever there is certain strains prevalent, we should have at least minimum 3 outbreaks and the complete sequence has to be mapped, and then we can declare it or conclude it, and based on that we can take a call. This is what I would like to highlight here. So you can see the GA24 lineage, which is circulating in India, the Indian local tribes, can cause the cystic oviduct cases in commercial layers and broilers, broiler breeders, and even some cases in broilers also we have seen, like cattle tracheitis, oviduct impactions, cystic oviducts, all these cases with nephritis. So then coming to some of the introduction of exogenous genotypes, how this happens globally and how it has happened in India. You can see introduction of the vaccine strains. Like in China, there was introduction of GA5 and GA6 were recorded in this particular country after the introduction of JAS and J9 vaccines from Australia. This is some of the best examples we need to learn. And GA13, that is a 4/91 like field isolates emerged in certain countries like Egypt, Chile, Costa Rica, and including India after the introduction of 4/91 or 793B in the Indian context, which is circulating in the Indian context, and we have found that. So the other example was the GA16, that is a Q1 strain from Europe to China, might be attributed to live breeder trade aimed at to enhance the local genetics. In China, it had created emergence of this particular strain in that country. But now from China, it has black flow to Italy also, and we are finding a lot of cases in Italy also with GA16. Okay. The other reasons for the exogenous genotype introduction is the migratory birds similar to the other viruses and the animal. Some of the examples of Italy. So emergence of novel genotypes in layers and breeders. You can see there are some of the isolates novel to the production drop issues, which we are finding in Indian context also, but not with these lineages, but with the lineages which we are having, like in layers and breeder, high mortality rate, decline in egg production, especially in the peak production. They are giving a lot of peak production without apparent respiratory signs and clinical signs. That's why people are not interpreting as any respiratory or anything, okay, which is seen in India also. So then coming to the recombination, how the things are happening. If you see, if when you are having two different viruses in a single bird, two different viruses enter a single cell, you can see a lot of recombinations, gene swiping can happen, okay, and lead to new variants emerging out. As such, it has a very poor proofreading capacity with the RNA polymerase, but when two different viruses are available, gene swapping can lead to the different. So these are all the different regions where the recombinations can happen, more susceptible, that is the non-structural protein sites and the spike protein sites also. Okay. So these are all the recombinant. This table shows you a lot of recombinant strains which are prevalent in different countries, which has emerged out. Okay. In different countries, you can see this is a country of origin in different countries, and these are the parental strains. But these parental strains, which has led to the recombination, mostly related to the vaccines, like here H124/19, H124/19. Majority, this is how the things are happening, leading to the recombinant sites. These are the genes. Okay. So this is one more example from China, where you can find a lot of isolates being emerged from 1958 to till now. A lot of new variants emerged out in this particular country. Okay. Of late, if you see, they are having so many isolates or lineages prevalent in that country, but more dominant is GI7, GI13, and GI19 is the most dominant. But of late, there is more circulation of GI28 and 29. If you see how the GI28 has emerged in that particular country, it is a multiple recombination event of these three viruses. It is H120, GI1 lineage, 4/91, and LDTT3. That is the local variant there. Okay, so the recombination of the field and the vaccine viruses had led to this particular emergence of new variant. Then the recombination, which has the new strain, which is GI29, you can see it is a recombination of 3 vaccine strains and 1 field strain. So this is how the things happen. When we are introducing different types of strains into a bird, it can lead to a lot of recombination happening in new variants. So this is what is happening in that country. If you talk about the recombination happening in India, you can see the earlier classification I had shown you. This is the recombinant viruses, where I had mapped it in collaboration with the CDAC. We had done that with the boot scan analysis. You can see that with the spike protein, there is swapping of the H120 portion mass type into the 4/91 type. You can see this is the H120, where the gene swapping at this particular position, 548 to 11515 base pair, that particular gene has been swapped. These are some of the recombinants, but I cannot say that this is the only recombinant which has happened. You can see a lot of recombinant viruses which would have emerged by this time. So, but the danger in the future is the diversity of this. This is some of the Australian publication. You can see this is the classical IB virus genome structure with the spike, 3B polymerase, envelope matrix, nucleic acid, all those things. But if you see the recent novel, some of the strains which have emerged in particular Australia, you can see there is deletion of this particular gene, 3A, 3B, and introduction of the new gene into the. So this is what is happening in some of the isolates in that particular country. So this is very important to know how the changes are happening with this particular. So then comes to the main thing, that is the vaccines we used as control, but we have seen a lot of we have learned in the last, can definitely learn a lot of lessons from that. Okay. But if you see here, none of these vaccines which were used during COVID was related to the live attenuated viruses. So most of the things are either inactivated or the recombinant ones, which has been used, which are much safer compared to the live attenuated vaccines for the introduction. But in poultry, if you see, we have usage of more live attenuated vaccines and inactivated vaccines. And we talk more about the protective types. By using two different viruses, we need to be having a broader cross protection. That is how the things we are trying to do that. Things. Let us try to understand about those things in the next slides. These are all some of the licensed vaccine strains available in the different parts of the world. That is, one is the GI lineage vaccines. If you see, in different licensed in different countries in different names. If you see the G1 lineage, the mass types, okay, M41 was the original strain, which in different companies 126. All those things. But European, that is, and the other one is the Connecticut. It is a mass variant. Okay, Italy 2886. The other major lineage, which is the 4/91 or 4/91 like strains, which are with the different companies in different names, that is 793B or 4/91, 1/96, all those things are CR88121. All these things are introduced. The third one is, which is more circulating, is the GA19, that is the QX type. Different companies, and in China, you can see different types of QX lineage and been registered. Korea. Apart from that, the major lineages, that is the local lineages, which is being registered in that particular regions, or this thing like GI12, the European countries, okay, with the Dutch isolates, D274 or 207 or D1466, or Delaware strain in US, GI41, that is GI27 in Georgia in US, Arkansas in US, the Australian, some of the strains here, you can see here, which has been registered. So these are all more local to that particular regions, more this thing. In India, we have GA24 as an inactivated vaccine. Okay. In this, both live and inactivated vaccines. But this particular strain, you can see introduction where homologic pathogenic variants are prevalent, then only it has to be. Otherwise, it is remains a controversial introduction. Let's see what is live attenuated vaccine strains circulating around the world. It is more of homologous vaccines applied in the field, leads to less variability. That is a known thing. We have to have a homologous vaccine, and so that you have negative selection pressure. There is no major changes can happen. So, but we need to have good lessons from COVID. You can see here, SARS, which was emerged in 2002, or MERS emerged in 2012 or 13, and the COVID. So people developed COVID. The vaccines were developed with COVID, not with the SARS or MERS, because they cannot cross-protect. We need to understand best example lessons from COVID. Okay, so we need to have a homologous strain so that negative selection pressure can. Non-homologous can lead to positive selection pressure. More changes can happen. And you are using a non-homologous, like you have GA24 in India. If you are using 4/91 or mass type, definitely they are going to have a positive selection pressure. A lot of variants can emerge out. And no vaccines is available. Definitely the variability will be high, but negative selection pressure. Only you can find that type of isolates when there is no vaccine. But that cannot happen, right? We cannot live without vaccines. So, but when we talk about homologous vaccines, there are some exceptions also. Like some strains are having 97% amino acid identity, but classified by virus neutralization, that is serotype, but they cannot have a cross protection. Okay, so even 2 to 3% variation can, if it is happening in the hyper variable region, that is the hypervariable region, they can lead to major changes in the spike protein in this thing and lead to less cross-protective. Okay, so like best example was Mass and Connecticut strains. They belong to the same group GI lineage, but they have a different antigenic. Even Mass and Butet strain, this is a best example. Conversely, there is some strains like they have a high variability, but when it comes to cross protection, they are very good cross protection. This is between the JP3 and JPQX. Yeah, this is very contrary to this particular state. So that's why always we should not go only with the genotyping. We should have both serotyping and genotyping classification so that you can identify a proper solution for both genotype and serotyping is very important. And some of the drawbacks with live attenuated vaccines, it can revert back to virulence. As I told, there is this particular RNA viruses doesn't have a proper proofreading. Sometimes it can revert back. The other thing which can happen is selection of minor subpopulation. So you can have a lot of quasi-species within a particular isolates. You can have different types of strains within that particular thing. So when we are vaccinating these particular quasi-species, which is very mild, they can start dominating and escape the immunity and lead to vaccine-derived strains and increase the virulence. And this can also cause genetic recombinations, and multiple live attenuated vaccines cause more virulent recombinant viruses. Okay? You have to note these points in a very careful way. The other one is immune escape mutant. You would have vaccinated in a proper way. But there may be certain strains which can have a selective pressure which are not neutralized by that particular immunity. They can escape the immunity and lead to emergence. Slowly, they can dominate over a period of time and lead to evolution of this. One example is the GA98 Georgia emerged with the immune selection exerted by the Delaware be used in the spread can be Sir, I was disconnected. Hello.
Yeah, now I think you went back to inactivated vaccine. Okay.
Yeah. So this is one of this thing where we can use, and you can include broader protection, including with the GA1 lineage 13, 24, which are circulating. We can update. That is the solution which you can have. That is some of the lessons from COVID also. Okay. So some of the vaccination programs, what I usually have experienced with this particular thing is go for early protection with the killed vaccines with all the genotypes prevalent. Okay, so you can go with the live vaccines on day old in the long-lived birds, then go for the inactivated vaccines at the earliest by the first or second. Then repeat this particular live vaccines by the 4th week with the mass strain only. Restrict your live vaccines only to one particular serotype or the gene lineage. Then that is the heterologous or something. And the killed vaccine should have a broad this thing with all this. Relay or this thing you can boost, and every frequently in commercial layers, maybe every 30 weeks, you can go for the repetition in the laying stage, and in the broiler breeders, every 18 to 20 weeks. That would the best way to protect. Repeated live vaccines leads to the emergence, which I have already told you. More circulation of this virus, more chances of mutations. So go for early protection of the oviduct with this particular schedule, and this gives a long-lasting immunity in the laying stage. Okay. So this is with the vaccine programs, which really gives the best result in the short-lived birds. When there is more circulation of such sort of lineages in the broilers in that particular area, you can definitely opt for. Normally, you go for a live vaccines with the MAS or H120 on this particular days or this thing. But when there is more circulation, endemic cases in that area, you can go with the NDIB multi or IB multi. Okay, with all these lineages prevalent, that is the MAS, 4/91 and GA25, which can give a proper protection. The live killed combination will be, of course, biosecurity, as it is a short-lived, you can not get a complete immunity in this particular cases. Other things has to be taken care of. So what are the vaccination challenges against emergence of new variants? I have already explained you. Okay, these are all some of the things, the heterogeneity of the IB variants or the co-circulation of different genotypes. All these things are involved in the emergence of new variants. So we need to have a proper track of this particular things. I have explained all these things in my particular slides. Okay. So whatever the vaccines you have, but if you don't have a proper vaccination programs at the field level, definitely it can lead to break of the immunity and break of failures can happen. So according to me, 99% the vaccination failures happen due to the vaccination programs, not the vaccines. Vaccines, strain variations, it is 1%. That is what I have seen with my experience with the spray vaccinations, or which is, if it is not uniformly, it will lead to circulation, cyclical infections and leading to prevalent of this particular virus. See, eye drop vaccinations, handling large number of birds definitely matters. Even the injectable vaccines has to be done in a proper care, has to be taken. That is what I always stress. Vaccination has to be done. So sometimes when it is not happening, leading to a lot of vaccinal reactions with the cyclical infections, which is from the vaccinated to unvaccinated. And also when there is other complications which is happening, like mycoplasmas in the farm or other bacterial infections or the viral infections in the farm, these vaccines can lead to a lot of vaccine reactions. This is how the things happen. Okay. So sometimes even I have seen some of the live vaccine strains, the new heterogeneous types you introduced into the broilers, you can see a lot of nephritis with the cystic ascites cases, nephritis, all these cases with high mortalities, 20 to 25% seen in this cases. Okay. Definitely take care of such sort of introduction of the new serotypes, and also have a good control on mycoplasma. You can see some of the published literature where the more problems with the MG and the IB. Whenever there is no MG free flock, definitely when you are using certain testing, there will be reactions. It needs to be taken care. The proper control of them over the mycoplasmas, that is what is required. So have a proper bubble to crack this particular. Coming to my last conclusion slide. So all this describes think globally and act locally, way of tracking the disease, what is happening, have a proper biosecurity, restrict the spread, which is very important. Identify the problem with new technologies, which is very important. Don't go with the postmortem only. We have to, because you can lead to a lot of confusion. Okay, so take the help of the labs and identify the proper strains. Definitely control the comorbidities, which is silent killers in these cases, and know your strains circulating properly. So proper sequencing of the genome has to be to know the variants, and then we have to have a, and we need to track it. That is not a end; it's a moving target. Proper monitoring of this particular serologies can also be done by HA and ELISA, and stop misinterpretation and abuse of live vaccines. Necessarily repetition of these live vaccines is not needed. You can go for a priming with some homologous in the initial stage. So definitely, it is a moving target for the vaccines. Vaccination does not prevent infection. Keep that in mind. We have learned a lot of lessons from COVID. Even after vaccinations, there were Omicron, especially Omicron. There was changes in the variants, but it can prevent the mortalities happening, but it cannot prevent the infection happening. These are all some of the good lessons which we need to know. Definitely, vaccination without biosecurity is a weak formula for protection, but biosecurity without vaccination is very unrealistic for disease. We need to have vaccination along with biosecurity. With this, we can definitely think locally and act globally too, which we can. So the last slide, which I want to correlate with our particular case, is the human polio virus, which also belongs to the single-stranded RNA virus, this is the recent published this thing in the newspaper, where you can see the polio causes due to the oral vaccines, which is emerging out, the vaccinal this thing, poliomyelitis, which is emerged. You can see some of the expert statement which was given long back, published in Lancet by one of our eminent scientists, Jacob John. Okay, so for achieving zero incidence of polio by 2000, the global this thing should have positioned inactivated polio vaccine in the middle income groups so that we could have phased out oral polio vaccine by this time. So which had led to the vaccine associated paralytic polio. So this statement is very important to be noted. We need to understand, we need to correlate with our infectious bronchitis. So thank you very much for this thing. I also thank the Avian Health Professionals Group, Association of Avian Health Professional Groups, for giving me this opportunity for highlighting about IV. Thank you, sir.
Thank you very much, sir. Participants, you can ask questions regarding this presentation, please.
Dr. Swati, we can defer this question answer session at the end. Now you can invite Dr. Kurkure. Dr. Swati?
Yes, sir.
Question answer maybe we can take up at the end.
Take up later. Okay.
After second presentation.
Thank you, sir.
Yeah.
Okay. Thank you very much, Dr. Prakash Reddy, sir. And now I invite our next guest speaker, Dr. Nitin V. Kurkure. Before he starts his presentation, I would like to briefly introduce Dr. Nitin Kurkure did his master's and Doctor of Philosophy in veterinary
see my
pathology.
presentation?
Yes, sir. Yes, sir. Right, sir. Right.
He started his career as a veterinary officer in 1991 for a brief period. Later served as a livestock development officer in Animal Husbandry Department, Government of Maharashtra, and then joined MUFSU as a teacher in the cadre of assistant professor and served as an associate professor and also the professor. Currently, he is serving as the Director of Research for Maharashtra Animal and Fishery Sciences University, Nagpur. He has many awards and published many scientific papers in international and national journals and handled many projects of DST and DBT. Currently, his research is focused on tackling antimicrobial resistance in bacteria of chicken origin with exploring the phytogenetic material and nanotechnology. Today, sir, Dr. Kurkure sir is here to speak about the subclinical coccidiosis and its control in poultry. I welcome Dr. Nitin Kurkure sir and request him to start his presentation.
Thank you, Dr. Swati, for a nice introduction. First of all, I thank AAHP for giving me a platform to share my views about the coccidiosis. Can you listen me properly?
Yes, sir. You are clearly audible.
Yes, sir. Yes, sir.
Okay,
Yes, sir.
thank you. So today I will be sharing my experience regarding subclinical coccidiosis. and we will see about its control also. This control part can be for subclinical and clinical both. So my outline of my lecture will be, we will revisit about the pathophysiology of Hymeria infections again. We all know, we have all wrote this in quite a few times in our undergraduate and postgraduate, but still we will revisit it. And then we will see the subclinical coccidiosis, how to identify and its prevention. It's just small talk. I will be limiting myself only more to the subclinical coccidiosis. Just have a recap of the what we learned and what we practice. Coccidiosis is a major parasitic disease. Since we started the commercial poultry farming and we have moved the poultry birds from backyard poultry to the cages, we have controlled all other many of the parasitic diseases like roundworm, tapeworms. They all have been controlled, but still we are struggling with the another parasitic disease, that is coccidiosis. In spite of having improved a lot on our managemental aspects and feed regime and everything, still we have problem. This disease is caused by typically 8 Hymeria species, which are listed here like Acerulina, Maxima, Mitis, everyone we know about it. Now we more worry about the Tenella, Nicatrix and Acerulina more now. So these are the 8 different Hymeria species which infect. In earlier topic, Dr. Prakash has very well elaborated about the IB, its variants and genotypes and many things. There are many other within the tunnel also there are many genotypes, but still at present we are struggling with the species, different species, and maybe genotype will come later when we control this. So again, when we think about its control, we have to remember few points which are very, very essential. Unlike bacteria and viruses, the life cycle of Hymeria is very complex. Bacteria, very easily they divide, they share split genetic material and they can divide into. Virus uses the host cells for its multiplication, and then they are released and then they infect the new cells. But Hymeria is something different. It is a very complex life cycle. It has asexual reproduction. It has a sexual reproduction, forms the oocyst, which are resistant to the environmental conditions. So it's a very complex. So as the complex life cycle is, that the more complex is the control. Then if you see the pathogenicity depends upon the species of the coccidia which is infecting. The disease produced by Hymeria maxima is different than Hymeria tenella. Hymeria nicatrix is different. Also, the genetic makeup of poultry bird is also very important. We have egg type of bird, that is long-lived bird, and short-lived bird, that is broiler birds. So if you see coccidiosis, the expression of the disease with the same challenge is more in the layer type of bird or long-lived bird. They are more as compared to the meat type of broiler. So genetic makeup is also very, very important. Whenever there is a frank outbreak, we have experienced high morbidity, high mortality. These conditions have, over the period of time in last 2 decades, has come down, except free range conditions where we can find the frank outbreaks and huge mortality. Anyway, I don't say it has been completely controlled. Still, it is there, but the incidence has been reduced. Then, point which is very important is subclinical. The partial control results into the subclinical disease, wherever it's a clinical or not. But when we use any half-hearted efforts to control the coccidiosis, it results into a subclinical disease, which is more dangerous. Also, if you see the immunity, like earlier talk was on immunity and how vaccines we can help. Immunity against coccidia is very specific. As it's a parasite, very complex structure, the immunity against Hymeria nicatrix will be very specific to nicatrix only. It will not be for Tenella. So this is how the parasite survives in the body, and these points we should take in our mind before we go for its control. Coming to the economic losses, it's a very, very important disease. After controlling the all other, most of the other infectious diseases, this is having a upper hand on the economic losses. Apart from the exact disease and morbidity and mortality, nowadays what we see, the more economic losses are due to the poor feed conversion in broilers and reduced egg production in the layers. It is not a huge, but the FCR is compromised. I have some my own papers or data to share with you, which I will tell. In USA, almost 18 years back or 17 years back, they have made a study, and they estimated the loss of around 3 billion US dollar 16 years before due to coccidiosis. And this economic burden was due to mortality, lower feed conversion, malabsorption, impaired growth rates, and reduction in egg production of layers and breeders. Adding to that, it was a medication cost. But this was 16 years before study. In India, we don't have such study. If it is with some companies, it is not been published. I know few of the companies, they have some data about it. Elanco and all those have data, but it is not shared publicly. So I cannot put it here. But I am very sure that it will match with the US conditions, and the losses are very, very high. And why I'm putting this fact here, because there are many participants from the industry. Unless you put this data on the record or publish somewhere, the government doesn't think much on it. The policymakers doesn't think much on it, and they don't invest in the research how to control about it. If it is hidden, it will go unnoticed, and the losses will continue, and research institutes will not pick up it in a right manner. So, coming to the pathophysiology, that was about the Eimeria coccidiosis. Now, coming to the pathophysiology, we all know about it. I will skip this, and these are the 8 parasites: Eimeria acervulina, Brunetti, Maxima, Mivati, Negatrix tenella. Each of these pathogen or Eimeria species has a specific site of infection. Like this is for acervulina, this is for Maxima, then again Mivati, this is for Nicatrix. And I have tried to adjust the size of the dots with the lesions produced. This is my own creation. The pinpoint hemorrhages, I have tried to make the small dots, and in Nicatrix, as the hemorrhages are very much pronounced, I have tried to make the dots very larger. So the organ specificity is there of these parasites, these Eimeria parasites, organ specificity is there, and also the type of hemorrhage that also varies. Like in Brunetti, it will be very, very mild petechial hemorrhages will be there. But if you see in Nicatrix, it will be huge, a larger quantity of the hemorrhagic spots will be there. So like this, the pathogenicity depends, and it is very site specific. On site, also you can identify that this can be this, but you have to make the differential by seeing in the microscope sporulation of these oocysts, and then doing the micrometry is the best way to identify what parasite it is. And nowadays, we all go for the PCR for the identification of these parasites. It's easy and perfect method to identify which type of Eimeria infection is there. Now, this is the life cycle. I told you it is a very complex. We all know about it, but just we will revisit it. So, when the bird ingests the sporulated oocyst, there is the sporocyst and sporozoites are released. They penetrate the intestinal epithelial cell. Again, there is a site predilection depending on the species which is there. It will enter into the gut epithelial of that particular site, and they will infect the cells. Once they infect the cells, the first generation scizones are formed, and the merozoites are released. They again penetrate the intestinal cells, and then they form the second generation scion, second generation merozoites and release of merozoites are there. These merozoites again come back and they infect, and this cycle continues. So from second also it comes back and again infect, and this cycle continues for 1 day, and it continues multiplication, so that larger amount of the second generation merozoites could form. Once the sufficient number of merozoites are formed, then the second generation merozoites, they enter into the epithelial cells and form the male gametocytes and female gametocytes. It is not like that only the bird has ingested, first generation has formed, second generation has formed, and next day there will be production of oocyst. There is one term in bacteriology, we call it as a quorum sensing. So it applies here also. After the second generation merozoites are released, it again infects the cells to have the larger population of these merozoites to be formed. Once the sufficient number of merozoites are formed, then only there will be formation of male gametocytes and female gametocytes. They fertilize, zygote is formed, and then oocyst is released. Oocyst is released and it sporulate in the environment. So this is very complex kind of structure, though the life cycle is completed within 6 hours, 6 days to 7 days. But unless there is a sufficient number of second generation merozoites are formed, you will find the oocyst production is very, very, very less. Very, very less. You won't find it in litter or in the droppings also. Though there is a disease, sometimes you don't find oocyst in the fecal matter. The reason is that the second generation merozoites, they again penetrate, again they circulate. They cause the disease, but oocysts are not. Now, a few things I have already shared. The pathogenicity depends on the ability of infection of this pathogen, and this depends on the type of the Eimeria species like Maxima or Mewati or Nicatrix or Tenella, like that. Replication capacity of the parasite. Eimeria tenella is the highly replicating parasite. And it is an estimated, theoretical estimate that one Eimeria tenella, if it is allowed to do the complete multiplication, it can produce 2.5 million of the parasites. From one oocyst, it can produce that much, if it is allowed. But there are a lot of restrictions on this. There is a host defense system. There are some microbiota there. There are certain bacteria which feeds on these merozoites or scizones. So there is a natural elimination of these parasites are also there. But if it is allowed, one can produce the 2.5 millions of the oocyst from a single. Afterward, followed is Nicatrix. Nicatrix also have very high potential of multiplication. But if you see Maxima is very, very less. Maxima cannot produce. So it depends on the replication potential of the parasite. And once the parasite damage the intestinal mucosa, there is a lot of pathology happening there. I won't talk much on that, what exactly happened, because it is very free, frank disease. Anybody can diagnose. Basically, there is epithelial damage, superficial epithelial damage, followed by underlying huge inflammation, and once the bleeding starts, you can see in clinical cases, there are a lot of hemorrhage, but in subclinical cases, you won't see the bleeding, or bleeding will be very minute, but because of pathology, underlying pathology, the height of the villus will be reduced, width of the villus will be increased, and because of the height is reduced, it appears, we call it as a villus atrophy, and the digestion is hampered, absorption is hampered, and all other things starts. So again, in pathophysiology, if you see and if you compare the clinical signs, a good pathologist or a good disease diagnosis or the person who has experience, well experienced in this, can little bit differentiate about the species on the clinical symptoms and the lesions. To tell you few things, watery and whitish diarrhea, if you see, it is a Eimeria acervulina. Dehydration, wet loss will be always there. And if you see some blood or bloody tinge, it is mostly the tenella. And if you find only mucus is there, high amount of the mucus is there, and thickening of the mucosal layer is there, it can be Eimeria maxima. but this needs to be too much practice or experience to do this, and the errors has to be corrected by laboratory confirmation. Once you diagnose something, let's confirm it with the laboratory, and this is how the experience builds, and then you can diagnose the species by the lesions and the symptoms. Then I told one thing I told you there, the about the infectivity of the host, that layer type of bird are more aggressive in the symptoms and lesions, and the disease is more in that. Also, it holds good that for the young chickens, they are more susceptible than the older chickens. Like this disease is mostly seen, particularly if you see in breeders or the layer type of bird, it is mostly seen pre-laying. In pre-laying phase, you will see it more than the post-laying. So young chickens, they are more susceptible as compared to the older chickens. They are relatively resistant. Doesn't mean that they don't suffer. They suffer, but relatively resistant, either because they are either immune with the earlier experiences, they develop some immunity, and that's why they don't show very frank disease. So these are few pictures which I tried to. This is a Nicatrix, typical Nicatrix pathology, and this is a Tenella pathology, very organ specific. Lesions are very different. If you open from inside here, it will be more petechial hemorrhages, and in Eimeria tenella, there are huge hemorrhages in the ceca, and you can differentiate it. One very important thing about when we think about the control of it, we must know about the how the immune system responds to the parasite. Whenever there is an infection, the two things can happen. One is the dose is very less, very less, and the parasite doesn't multiply to that extent. Like first generation silent forms, second generation silent forms, and good population of the second generation merozoites are not formed. There will not be any infection, and the bird will be healthy. And if infection happens, again there can be a no disease with the looking to the it went into the subclinical form. Apparently, there will not be any disease, or if the sufficient number of the second generation merozoites are formed, there can be a disease and the hemorrhage, and this will start. But anyway, whether it is a subclinical or a clinical, there will be acquired immunity to this bird, and the bird will be little bit resistant for the future exposures. So low level of exposure to the coccidian develops into a protective immunity. And Eimeria maxima, though it produces very low number of oocysts, but it is very good as compared to the, if you consider the immune response, as compared to Tenella or Nicatrix, Acervillina, maxima produces the highest immunity as compared to other. But this immunity against maxima is only for maxima. It is not usually shared with the other species. So that is a limitation, but it's a very good immunogenic as compared to the other coccidial parasites like Tenella and Acervillina and Nicatrix. The early development, I have told you in the life cycle, there are asexual reproduction stage, sexual reproduction stage. So early developmental stages of coccidial life cycle are more immunogenic than the latter or sexual. During the latter part, the damage is more, inflammation is towards pro-inflammatory. So it is more towards the inflammation, more gamma interferon production, more disease is there in the latter stage. But in initial stage, more the T regulatory or T helper cells, they are triggered and they help in production of immunity. So early stage of coccidial life cycle are important for the immunity development rather than the latter part. And Eimeria maxima have a greater immune response potential as compared to other Eimeria parasites. So because of this pathophysiology or what are the pathology happening, there can be a clinical disease which easily can be diagnosed. Mortality can be huge, blood in feces, ruffled feathers, diarrhea, loss of body weight, huge. Easily we can identify this, and then it can be treated accordingly very nicely. But worry is the subclinical coccidiosis. It is very, very prevalent all over the globe. None of the country can say, okay, they are free of it. Like we veterinarians, when we teach, we tell that few of the continent, they are free of rabies. It's not for coccidiosis. Coccidiosis is everywhere on this globe, observed in the more size of the flocks. The flock which are of the higher sizes, the chances of the subclinical coccidiosis is high. Like giving comparison, if the flock size is 5,000 and flock size is 25,000, the 25,000 flock size will have subclinical chances of having subclinical coccidiosis in 25,000 flock is more as compared to 5,000. And for the incidence of the subclinical coccidiosis, there is no remarkable difference for the any coccidiostat used, whatever the broiler strain is there, whatever the feed type is there, and feed composition is there. It is more with the management and the flock size and all other things, rather than the uh, genetic potential, coccidiostats use, and uh, feed composition. This is a very good paper published in 2015. Subclinical coccidiosis is also a result of over and long-term use of coccidiostats. So same which is happening for the bacteria, we are witnessing the AMR in bacteria. Same is happening with these parasites, and these parasites are more equipped than the bacteria for development of resistance because of their life cycle. Their life cycle, some stages happens outside the body or outside the host. Some stages happens inside the host. In the host, there are two type of different multiplications are there, so they have ample chance of changing their genetic potential. So over and long term use of coccidiostat has lead to development of drug resistance, and they are responsible. Mostly these drug resistant uh, Eimeria strains are responsible for subclinical coccidiosis. And most prevalent, if you see, the most prevalent subclinical coccidiosis species are Acervillina and Tenella, because these has the highest potential of multiplication. When they multiply, each time they have a chance to make the change in their genotype or their genetic makeup, that or acquired few more things, bringing change in them. So as their reproductive potential is more, they change fast and they lead to the drug resistance and subclinical coccidiosis in poultry. What are effects of this subclinical coccidiosis? Mostly it goes unnoticed. Mostly it goes unnoticed. The subclinical, it gives rise to the highest proportion of economic losses by poor feed conversion in broilers and reduced egg production in layers. Poor feed conversion, I have some data I will share with you. But in egg production, there can be reduction in egg production 1 or 2 or 3%. Not much, maximum 5% because of subclinical coccidiosis. Nobody notice it. Or if it is there, some other things are blamed for it, or even the attention goes to the other things, and coccidiosis continues on the farm and it continues the losses. So as it is not been thought properly, or even it doesn't come in the thinking of the diagnosticians or the veterinarians, those who visit, he tries to hit the other causes, and the treatment is given for other causes, and coccidia enjoys the small subclinical stage and produce the disease. The losses aggravates as the bird receives treatment for something else. Any treatment which is given to the bird is an additional burden to that bird. Additional burden. It has to metabolize that treatment, whether it's an antibiotic or something, antimycoplasma, anything which is given, it's an additional burden to the metabolism. Because of this, there is a chance for this Eimeria pathogens to multiply faster and produce the disease. These are two studies. One is these are 2008 studies from Ghent University, and they have estimated in 2008, 0.1 euro, means almost, I think, 8 rupees in Indian currencies, is loss per bird. This is the loss per bird. And recent study in 2020, they have estimated it 0.18 euro is almost 15 rupees per bird loss because of subclinical coccidiosis. These are not really means thought, but they are there. And in India, situation is not different. I'm very sure subclinical coccidiosis is there on each and every farm who has a capacity above 10,000. It is there. It has not been looked. It has to be looked. All will agree with my statement. This is our data in 2009. And from actually, we started working on coccidiosis in 2004, having some alternative approach, and that's why this is a data which we have. At that time, this is very old data, 13, 14 years back. And because of subclinical coccidiosis, there is a difference of the weight gain is about 90 grams up to fourth week. And another data, recent data, is my master degree student, Dinesh. Then we thought, we planned this, and we did the comparison, identification of subclinical coccidiosis and then treatment. And you could see by the end of 6 weeks, there is a difference of uh, normal flock and subclinical coccidiosis affected flock is about 300 grams. And when we noticed it at third week, and then we tried to treat this flock, and then, of course, the bird achieved the body weight, but still they were deficient of around 150 grams. So we could make, after diagnosis also, we could make it out. We could cope up only 150 grams, but still there is a loss of 150 gram per broiler. Per broiler, this is the average body weight. So per broiler, there is a loss of 100 to 150 gram because of subclinical coccidiosis. And here we use the alternative methods to treat it. Probably if we would have used the therapeutic regime like amprolium or other, it would have been slightly more, but this time we use some probiotic strains to treat the subclinical coccidiosis. So that's why the result is not very convincing. If we would have used any chemicals, it would have been far better. So that was about the subclinical coccidiosis and how serious it is. I guess everyone will agree with me, if there is a loss of 100 or 150 gram is too much in today's context. The rates are very poor for the lifting. Even whatever we are selling, if 100 gram less is, it is too much loss. And how to identify this? This is mostly encountered in broiler farms as compared to layer farms. You find it is more in broilers. Difficult to diagnose, and any diarrhea with or without bleeding on the farm should be suspected for subclinical coccidiosis. Any diarrhea, I'm saying. Diarrhea may be because of any feed ingredient or whatever the farm personnel or the owner thinks, whether it is of feed ingredient, whether it is of E. coli, whether it is of salmonella, whether it is Clostridia, whatever it may be. But it should be suspected for coccidiosis. This subclinical coccidiosis will invite all other problems too. So if we control this, probably the other problems can be also controlled. So any diarrhea on the farm with or without bleeding should be suspected for subclinical coccidiosis. And diagnosis of subclinical coccidiosis is not very expensive, very easy. You can diagnose it. On necropsy, if you see, there may not be free and frank lesion as that of clinical coccidiosis, but you can see some white patches, transverse lining on the outer or inner side of the gut. Minutely, if you find, there can be multiple pinpoint size hemorrhages on the gut that you can see. If you wash the gut, you have to, once you cut the gut, you remove it, the content, wash it with the water. Then only will you appreciate the subclinical coccidiosis lesion. Otherwise, not. If you are not washing them with the water, you are going to definitely miss the lesions of subclinical coccidiosis. So once you open the gut, remove the intestinal content, wash it with the gentle wash with the water, and then you will appreciate the lesions of the subclinical coccidiosis. Apart from that, there can be, if you are lucky, you can see the segmental ballooning or enlargement and orange stained mucosa. If it is a little bit severe degree of the subclinical coccidiosis, then only you can see segmental ballooning as well as orange stained mucus in that. But the best way is you can find the oocyst in the scraping or fecal sample in the microscope, and you can find it. Any person who can differentiate this is not air bubble can diagnose coccidial oocyst. It is not difficult. The graduate person can also identify subclinical coccidiosis on the farm. Just take the fecal sample or the litter sample, allow it to sediment and put it under cover slip and observe. You can very well, if it is subclinical coccidiosis, you can very well find it. But you should have patience to find it for them. If you have patience, definitely you can find, particularly in broiler rearing period, you have to visit between 20 to 25 days of the age of the broiler, have some fecal sample or litter material. You will find it if there is a subclinical coccidiosis. Very easy to diagnose, no hard rocket science in this. Use the sediment, put the drop, put cover slip, and you can observe it. No need of oil immersion. 10x, 40x, 20x is enough. Now, after diagnosing it, what can be best we can do to control this coccidiosis? It is very good to say good husbandry practice plays the role, but it is very, very difficult statement. Husbandry practices, there is no limit to it, how good it can be, but try to do it as better as possible. And major things in that, reduce the access of the birds to the feces. This should be reduced. And this mostly happens when the feed is not available throughout the day or life period of the birds. They try to pick the feces or the litter, and this is how the oocyst goes inside the body of the bird. Maintaining the litter quality, controlling temperature, ventilation, moisture. These are the key points for the sporulation of the oocyst. As far as the oocyst is unsporulated, doesn't matter how many oocysts are there in the litter. They are not infective unless they are sporulated. And these temperature and moisture will make them sporulated. So you have to maintain it so that there will not be sporulation, and moisture should be controlled so that there will not be sporulation. And thorough cleaning between the flock. This is not the new thing I am telling you, but still India is a very diverse country. We have certain area where every batch there is a clearing of litter, and we have certain area where the broilers are reared twice or thrice on the same litter. So thorough cleaning between the flock is recommended. Wherever possible, we have to do it. Coccidial oocysts are extremely resistant to disinfectant. Once they are sporulated, they are resistant. Routine disinfectants, what we used, they are not able to kill this sporulated oocyst. So these things also should be kept in mind. And most important thing is coccidial oocyst resides in the cracks and crevages. This is a very good article published. You can visit this article. You will find some more clues for the control of this about the subclinical coccidiosis in the article which I have mentioned it here. So basically, these coccidial oocysts, once the litter is cleaned or the farm is cleaned, mechanical cleaning is okay, done. Washing is also done, but these oocysts, they remains live in the cracks and crevages, even in the utensils which are used for the husbandry practices. If there is a dirt, they are microscopic, they are present on the dirt. So these sporulated oocysts, once they remain there, they will be ingested by the birds. The life cycle will continue. From one oocyst, there will be at least 1,000, not in millions, but at least thousands there will be produced. Again, these will be picked up, and this is how, may not be in the same flock, but next flock, definitely there will be subclinical coccidiosis. So for the control, this is very important. Coccidial oocyst resides in the cracks and crevages, and the dirt remain on the farm after cleaning. And there, these oocysts, they reside and they come in the broiler rearing once you put the broiler there. So now for looking for the coccidiosis control tools, the coccidiostatic drugs only prevent the replication. They are not for the complete killing. And the coccidiocidal drugs which we use both of these, they have complete killing action. So we should not be confusing with these two terms. We should know when we are using coccidiostat, our aim is to prevent the replication, but it is not completely killing or sterile effect. And when we are using coccidiocidal drugs, mostly chemicals, it is the it is expected to produce the sterile effect. All will be killed. Inophores, they are maximally being used, and that's why there is a maximum resistance has been documented till today for the inophores. They are coccidiostats used for the retarding in the replications of the parasite, and they are extensively being used. That's why you are finding some resistance to it. Synthetic drugs, which we use mostly in common words, we use the chemical coccidiocidal drugs. They have different mode of action. We will see how they are different. And this is the background for we should have in our mind when we go for the control of the coccidiosis. For the control, we have in therapeutics, we have sulfonamides, ultrazuril, amprolium. Amprolium and sulfonamides, they are mostly been used. They are wonderful. Current disease control programs are on anticoccidial drugs, mostly inophores and these chemical coccidiostats that we use. Some live vaccines are also being used for breeders and layers. These are the current disease control programs that has been used, but still we have some issues. So we have to think for the proper control of coccidiosis. So use of chemical inophores in combination or essential oils or other phytomolecules which are very, very good in control of these coccidial subclinical coccidiosis, that should be used, and use of different vaccines. Just now, Dr. Prakash has mentioned that live attenuated or the some live vaccines, they have some reversal effect. They can come back to a infective stage and produce disease. So we have to think of some different type of vaccine approach for this control of coccidiosis. So among the anticoccidial, we have typical inophores, that is salnomycin, monensin, narasin, maduramycin, lacellucids, all have been used. Wonderfully, they were doing so far. In last 2-3 decades, they helped us very good. Chemical coccidiostat, rominidin, which has been mostly been used, daclazuril, nicarbazine, they are doing very good. Now their use has been increased. Also, there are certain companies which has these in combination, like inophores and nicarbazine mostly. They are being used, and they are also very good in this. So far, these are the anticoccidials which are available for us for the control. We will see how the inophores act. Like basically, inophores, as the name indicates, it is an inophore. So they take the ion inside the cell. Like this is the outer surface and this is the inner surface. The ions, mostly sodium or potassium, they are brought by these ionophores into the cell, and in the cell the ion concentration increases. Once the ion concentration in the cell increases, there is a rupture of cell, and there is a certain mechanism of bringing them inside and sodium from inside to pour outside, that is sodium pump. But the parasitic wall is a hydrophobic wall. So in hydrophobic wall, these ions, when they come, they translocate. There can be some damage to this. But ionophores, they are just like they made these hydrophobic. They cover these ions and they bring these ions inside the cell to this hydrophobic membrane. The ion concentration increases, and then there is a swelling of the cells, and the cell disrupts. This is basically this is the mechanism also used for the bacteria. But these ionophores, what we sodium and potassium ionophores are there, we use for coccidial control. The other ionophores which brings calcium and all, we use for the antimicrobials. So there are different ionophores, like some brings sodium inside, some bring potassium inside, some brings both. Mostly we call them as a divalent cations. they are divalent cations. Maduramycin is the best example. So they are divalent cation. Sorry, Lacellus is the divalent cation, which brings both sodium and potassium. So the reaction is faster. And then they break the scisons, merozoites, and the cycle is blocked. So if you see the monovalent ionophores are the salinomycin, monensin, and narasin. Trivalent is the lacellucid, and glycoside ionophores are the maduramycin and semduramycin. So these are their mechanism remains the same. As monovalent is just sodium. This will bring sodium, potassium. So as their mechanism is same, so if the Eimeria oocyst is resistant to salinomycin, automatically it will be resistant to monensin and narasin. So once you are using salinomycin, and if it is resistant, there is no fun in using narasin or monensin. Same is the case with the maduramycin. If you are using maduramycins for a long time, and if there is a resistance, there is no use of using the semduramycin. So there is a cross resistance, cross protection against them because the way of mechanism is same. So when we think of the resistance and control them, we have to use the different chemical or the agent which has a different way of mechanism of action to control this. For this, we use the chemical coccidiostats, and in that there is a nicarbazine. Nicarbazine has a different way of action. Basically, it paralyzes the intracellular ATP synthesis. It paralyzes the intracellular ATP synthesis. When ATP is not synthesized properly, the sodium pump doesn't activate, and then there is a retention of the sodium ion inside the cell, and then the cell disturbs, ruptures, and this is how the control. So here, it is a bit different from the ionophores. It acts on the ATP synthesis. And if you see the robinidine, which is now mostly been used in the pre-starter diet, it inhibits the oxidative phosphorylation. Again, same, it reduces the ATP synthesis, which is required by the mitochondria. It works as an energy house. And this ATP synthesis is hampered, but here the mechanism is something different. Here, the mechanism of reduction in the ATP synthesis is inhibiting the oxidative phosphorylation. So this is how they have certain different kind of mechanism of action. So once it is resistant to the ionophores, you can use these other chemicals as their mechanism is different. The oocyst or the scisons or merozoite doesn't have a mechanism to overcome this, and they die, and we can control the coccidial oocyst. What happens exactly when we use the same coccidiostat for a longer time? Suppose this is the population present on the farm. Every farm has a coccidial oocyst population. Don't think that my farm is sterile and it doesn't have. Every farm has a coccidial oocyst. So this is a population which is available on the farm, and in that there are only two oocysts which are resistant. When we are using the anticoccidial, what happens? The coccidia, whatever the anticoccidial we are using, it will kill the sensitive oocyst, but the resistant population will increase in the flock. Next flock, again next flock, again next flock. So after certain few flocks, maybe 3-4 flocks, you will have population of only resistant coccidia species. And on the same farm, if you are using without any anticoccidial, there are chances that only the sensitive parasites will, or this Eimeria will multiply, and we can eliminate this. So I don't recommend here that you don't use any anticoccidial. I'm just telling you here the mechanism, how the resistant Eimeria parasite survives on the farm and their population increases. So to control this, or to control the subclinical coccidiosis, what best we can do? In India, we have certain flocks where the same litter is used. So in that flock, try to replace the old litter. We have to convince, if it is economically feasible, best way is to change the old litter system. change from ionophore to the synthetic drugs, you have to change the type of the drug which you are using for the control of coccidiosis. And if possible, vaccine should be used. Vaccine is expensive, I know, but vaccine should be used where you do not have liberty to change the liter. You have to think for the vaccines which can be implemented. Or you can use the shuttle program. Shuttle program is nowadays used by almost all farm. I think in 80% of the farms in India, they use the shuttle program. So that is what we use two different medicines of the or anticoccidials in the feed. We use some chemicals in the starter feed or ionophores in the latter phase, like a finisher feed or something like that. Earlier, we have straight program like using ionophores from pre-starter to finisher. Now we are using shuttle program, and we should think of something else, that is rotational program also. So shuttle program, we can change, we can have the different anticoccidiostat during the different phase of feeding. That we can do that in chemicals coccidiostat we can use in the pre-starter, and in finisher feed we can use some different, that is the shuttle program. Within that also, there should be internal change that we should do. If we are using ionophores, from monovalent, we should go to the some divalent sometime, and then again we can change to glycosides. So this change has to be made. If we go for rotation, then the meaning is totally different. Rotational control is you have to change the coccidiostat for that batch. Like first batch you are using ionophores, second batch you use chemicals. Or two batches you are using ionophores, third batch you use chemicals, again two batch you continue with the chemicals, next batch you again bring the ionophores or a herb, essential oils or herbs or other control mechanism. This is the rotation system. So we have to change the mind of the parasite, that if he is preparing for the ionophores, we have to give the surprise of the chemicals. If he is prepared for the chemicals, we have to give him surprise of the ionophores or essential oils. Like that, we have to change. So shuttle program is within the same cycle of the broilers during the different phases of the growth, and rotational program is different anticoccidial in the whole life of that broilers. So that this is a wonderful way. So far, we have adopted shuttle program, but we have to think of the rotational program also for the control of the coccidiosis. Then next approach is the vaccination. Most common in layers and breeders. In breeders in India also, many of the breeders, they are being using it, but not very common in broilers. Live anticoccidial vaccines, they are better than the first generation, which was very wild type. The present vaccines, what we have is live attenuated. So far, so they are working well, but still we have to find some even less pathogenic way of the producing vaccine, which will produce the enough immunity, but the multiplication of the parasites will be less. No doubt they are very high cost and product. There is another limitation of the live attenuation is the production capacity is limited. This is not a bacteria or virus that you can go on pretty plate or tissue culture and then you can produce the as much as doses you want. For producing this vaccine, you have to have the living system, that is birds there. So there is a limitation. So that's why we need to have some recombinant kind of things or some different kind of vaccines. Dr. Lile Hodge is working very nicely on it since last 25 years. She's working, and probably she is having some candidate vaccines available with her for alternative approaches for the vaccine. Maybe it will take some time, but in India so far, uh, I have some information. Nobody is working on it, but I put these questions even to the industry or even the academicians, those who are attending. We should think on this because it's a problem and we have to find the solutions. Mechanism of vaccine is again very simple. Vaccination is replacement of the strain prevalent on the farm. It is nothing to do something to control or to kill the parasite. Mechanism is very simple. If you have by using continuously anticoccidial in your farm, you have the highest population of the resistant strain. By using vaccine is attenuated, so it is less pathogenic. So using vaccine, you are increasing the population of less pathogenic strain on the farm. Doing vaccine for the Eimeria doesn't mean that you are eliminating Eimeria. It's not. Vaccination is replacing the resistant strain or pathogenic strain with the milder strain. It's only that. That's why I say current approach is this, but we have to think of something other way of vaccine production so that we can reduce the population of whether it is a beneficial, whether it is a pathogenic or less pathogenic, but we have to reduce the population on the farm. So the challenges what I feel in coccidiosis control measure is inophores are considered antibiotics in many of the developed countries. Like Europe, it has been considered already MRLs are set for the different inophores and chemical coccidiostats. Chemical coccidiostats has a longer withdrawal period also. Inophores are considered antibiotics. Slowly it will come to India also. If we think to replace this, we are thinking of vaccines. Vaccine doesn't have all strains in it. And I told you, immunity is very, very strain specific. Immunity to maxima will not be applicable to Tenella. In a when you see the vaccine dose of the for the coccidiosis, you won't find all strain included in that. So, and most of the vaccines, they are imported from outside the India. The strain which are prevalent here, we have to attenuate and use for our own farms. And development of the resistance to inophore is the alarming. That's why these all issues are coming up. We have to think something different. So there are some alternative approaches also available for the coccidiosis, that is use of some enzymes. This is what we can say repurposing of the drug, what we say enzymes. Certain enzymes has some activity which can reduce the coccidial multiplication in the gut. Probiotic, I told you Dinesh, one of our student in 2016, he worked on it. There are some good probiotics which can control the coccidial multiplication. Basically, these bacteria, they compete with the nutritional requirement of the scizones or merozoites, or even some bacteria are pathogenic to these merozoites and they kill them. Essential oil, herbal essential oils are the very, very good, and they are very, this is the thing which you can count on much more for the control of coccidiosis. So by this, I think by using this, you can inhibit some effect of this, improve the gut immunity and these biases also. So these are very good alternative approaches available with us. We can think and use it. So basically, these phytochemicals always have some questions raised. They do not work as per the chemicals or the synthetic drugs. Definitely, they will not, because the way they were used till date were different. But now these phytochemicals, they have been in purified form. Their activity can be increased by using by changing their properties using phospholipids, even nanoparticles which can be made. That is what the area where we are working. And so far, to our experience, they are wonderful. So there are these phytochemicals also has some prebiotics in it, which helps the probiotic bacteria to grow. And these probiotic bacteria can be pathogenic to some of the Eimeria species developmental stages. And this is how they can reduce their population. Along with that, they have anti-inflammatory activity, immunomodulatory activity that can be of useful to us, immunity in enhancement. And secondly, few of the phytochemicals we have found or even documented, though the oocysts are formed, these oocysts are not sporulated. These oocysts are not sporulated, or sporulation capacity of the oocyst is compromised. So the next challenge to the next bird is reduced. So to summarize these are some essential oils I am showing you. they have activity, most of them have activity on sporulation. By feeding of these essential oil to the birds, along with when the parasites it's multiply inside the gut, their sporulation capacity is lost. So this is wonderful way they can act. Also, when they have the good, they improve the immunity. The first generation Sison, first generation merozoites are released, but second generation merozoites are not released. So there the T cell immunity or cell mediated immunity acts very nicely. Curcumin, carvacol are wonderful in this way. So multiple essential oils are there. In combination, they are the molecules which can be used control of this. And the beauty of it, they don't have any withdrawal period. When whatever the food is going to the human consumption, they are quite safe. Usually considered as a safe, so that they can be used, and we can count on this. So, I will summarize few by how to control the subclinical coccidiosis to minimize the losses. So, dry cleaning, removal of litter, feed residues and manure after every batch of the flock. Just think of this. Probably where the litter is not expensive, we have to do this. People are doing it, but where litter is still expensive, we can think of this, how it can be reduced. But unless we do this, the coccidiosis is not going to be controlled. Clean drinking lines, cups, reduce the subclinical coccidiosis. Definitely, whatever the litter material is not only on the floor of the shed, but whatever is going on the walls, on the drinkers, on the cups, and if they are remaining there, it will contribute for subclinical coccidiosis. Use of high pressure cleaners and detergent, it improves the efficacy of the cleaning. Nowadays, a lot of equipment has come. Even some equipments, they are advocating it to use for the cleaning. Definitely, they are of some use. We don't want any oocyst remain on the farm so that it will be infective to the birds which will be placed after it. And fill up the cracks on the floors and the walls, because the coccidial oocyst reside there. And once the oocysts are sporulated, it is resistant to disinfectant. So though you do the disinfection, you will be in the false safety that I have disinfected, but oocysts are remain present there. Okay, this disinfectant can kill bacteria, can kill virus, but oocysts are resistant. So this is one of the way. Actually, to tell you that we are working on one aspect, that we don't want any anticoccidial to be added in the feed, whether it's a chemical ionophore, nothing. Give it attention on the disinfectant. We are working on the disinfectant which can kill the sporulated oocyst also. That disinfectant, if we use on the floor, and then during the lifespan of the broiler, just use anticoccidial stat, that is essential oil or phytochemical anticoccidial stat, only twice in the life of the birds, just for in a gap of 5 days, and completely no need of giving any anticoccidial. We are working on it. Soon we will publish that data, and it's a very good approach for removing the anticoccidial or any chemical anticoccidial in the diet. So subclinical coccidial adopt shuttle program. If possible, you can think of some rotational program also. On the farm, remove old litter, change from ionophore to synthetic drug and coccidiosis vaccine. So these are the ways I have already explained about this. So these are the ways about the coccidiosis control. Then diarrhea with or without blood in the feces should be suspected for subclinical coccidiosis. As soon as you see the loose droppings or little bit litter is going wet as compared to 3-4 days before, suspected for subclinical coccidiosis. It is not at all expensive. Collect the fecal sample or litter sample, examine under the microscope. If anyone wants to test it, and if he doesn't have facility, just ship it to us at Nagpur Veterinary College. We will do it. We will do it very happily. Screening of fecal samples for presence at regular interval, I suggest any time between 20 days to 25 days of the broiler, you have to test it. And for layer flock, you have to do it intermittently whenever there is a little bit diarrhea you are seeing, or whenever the population of insects, beetles, and ants has increased on the layer farm. Just after that, collect some litter samples and send or test it in the lab. Any lab can do it. It is nothing rocket science. Anybody can test it. So test it. If you find something, do the lesion score, gut lesion score. Once the fecal sample are positive, open few birds, do the lesion scoring, and then you can treat the flock accordingly. The production will not be compromised. So I stop it here. I think I have the time has been over now. We are already delayed by 15-20 minutes. So thank you. If you have any questions, I will be happy to take it. Thank you.
Thank you very much, sir, for sharing the insights regarding the subclinical coccidiosis, especially about the elaborated control measures. Now I request participants to ask questions regarding this topic. Unmute yourself to ask the questions or type your questions in the chat box, please.
Good morning, sir.
Yeah, please
Hello.
go ahead.
Sir, is there any specific seasonal coccidiostat, sir? If it is summer season, we have to start with this type of coccidiostat, or if it is a winter season, we have to take this type of coccidiostat. Is there any restriction, sir?
Okay, shall I answer?
Yes, sir.
Yeah, it is nothing like a season. I
Yes,
told
sir.
you, coccidiosis control is a farm, or basically it is a shed management. So it depends on the which coccidiostats you are using in the earlier period, and the problems if you have, then you need to change. It may be in any season. Coccidiosis is not limited to the only rainy season, what we usually think. It's not limited to that. Only where the moisture content is high and temperature is bit more, it helps in the sporulation. So you see the incidence is more. But you can see coccidiosis in summer also. I have recorded cases of coccidiosis in summer also. So change in the coccidiosis depends on earlier coccidiostats which you have used, the program which you have used. And if you have problem, you have to change the group of the coccidiostat. You have to change the group of the coccidiostat.
Thank you, sir.
So there was a question in the chat box. Is the essential oil or phytochemicals are effective than chemicals or ionophores?
See, definitely the synthetic drugs, they are, as far as efficacy is concerned, they are more efficacious than the essential oils as on now today. Now we are working on their increasing their potential. How to we can increase the potency of these drugs. So that's what I told you. If you use the raw essential oils, their effectivity will be compromised. But if we improve its efficiency by making it microencapsulation or microemulsions, even phospholipid conjugation, their activity can be increased. And last year, our one of the student who did parasitology student who did research means by phospholipid conjugation, they are equivalent with the ionophores.
Thank you, sir.
Sir, nowadays farmers have started testing OPG counts in broilers. Can you throw some lights on what is the ideal OPG count during their life cycle in broilers, sir?
Yes, sir. Thank you. It's good to listen that they have started doing OPG at a certain age, the broilers. Very good. There is no thumb rule that you have this much OPG, then your flock is safe. Only thing, the OPG is high, like if it is going above 1,000, it is an indication that if today 1,000, after 6-7 days, it will be like 50,000, and you can have a clinical disease.
Okay, sir, understood. Unless you follow, there is a trend of increasing count, we should not be worried much.
Yeah. And see, the OPG will, with my experience, the OPG will be higher till 28, 30 days.
Okay.
And then it reduces because by that time the broiler develops its own immune system to control it.
Yep. Yes, sir.
Yeah. And even the microflora get changed. During the early age of the broilers, microflora is different, and latter part, microflora is different. So latter part has more pathogens in the digestive system. So because of that, even that bacteria, they are harmful to the scions and merozoites. So the population gets reduced.
Right. Thanks, sir. Thank you.
Hello, sir.
Yeah.
Yes, this is a general question for the welfare of the poultry industry as a whole. Generally, there is not much combined vaccine of viral bacterial together, so that we can avoid too many shots to the poultry, particularly in layers. Why that kind of effort is not taken by the scientists in India or abroad?
Yeah, good question. Uh, we have done so far for the viruses, multiple combination of the vaccine means pathogens against which vaccines can be developed. Bacteria, virus, and parasites, if we pull together, we have to think for the processing capacity of the host. If we
Yes.
are injecting some antigen to it, every host has some capacity to process it. Unless
Yeah, of course.
it doesn't have a processing capacity, you will give vaccine, you will be in the false assumption that I have protected my bird. But bird
Yeah.
has not processed that antigen, antibodies had not been produced. And if you see, the poultry has its own limitation. They doesn't have immune organs as the mammalians has, like thymus they don't have. Sorry,
Yes.
thymus these birds have, but other
Thymus.
organs they do not have. So bone
Yeah.
marrow has to work, even intraepithelial lymphocytes has to work. There is a lot of pressure on the immune system as far as these broilers are concerned. And apart from that, their own growth is there. That is one of the factors. So if we give too many antigens, diverse antigens, probably birds will not process it. That is the uh, thinking process which has prevented the scientists to develop such kind of vaccine.
Yes, of course, it's not possible to combine all prevalent viral bacterial infection together. What I mean to say, wherever possible, at least
I'm
that
finished.
kind of study can be, of course, with due experimental studies to prove the efficacy. All these things should be taken up. Yes,
Sorry.
sir.
वो बोल रहे हैं कि मेरे को बाद में खाना खाने का टाइम नहीं मिलेगा घर में मैंने कुछ खाना नहीं बनाया
Sorry, can you repeat? I was actually I missed
okay.
your question.
So what I mean, as you said, sir, it is not possible to combine all major pathogens together. But what I mean, the possible combinations can be tried, attempted. That's what I mean to say. So to prove efficacy by doing different experimental studies, and then say whether they could protect with challenge studies, all these things. Once it is proven, the possible combinations can be, advocated to the poultry industry. That's what I mean. So that kind of research can be initiated by major institutes working on poultry biologicals.
Okay, thanks. Good feedback to us. We will think on it.
Can I answer your
Yes, yes.
question, Manu? Yeah.
Yes, sir.
See, there are certain combined vaccines also which are available, which have been proven
it is
also,
that.
like
Yes.
ND, IBD, IB, and Rio virus vaccines. There are combined vaccines which have proven to be effective in the long birds, and which has been used in the market since several years.
Yes.
There are there.
My concern is some more can be added up. That's what I meant to say. Supplement, yeah. Anyway, thank you, sir.
Yeah, as Dr. Kurkure Saab has highlighted, there is certain limitations for those things.
Yeah. Okay.
Sir, already I put this question in the chat box, sir. Sir, if there is a watery albumin, so is it the reason for only IB virus from IB virus infection, or we can suspect any other
That
case?
would be the first. That would be the first suspect, which is watery albumin is one of the indications of the IB virus infections. Okay.
Yes, sir.
So definitely you can suspect. I would definitely go for a prophylactic vaccination rather than a therapeutic vaccination. So people do use a lot of live vaccines in the laying and all, but that doesn't really have a good picture. Okay, you are adding more viruses into the environment and into the bird for that to process effectively. So for certain support use you can give, and later on you can go with the inactivated vaccines. That is the only this thing, so that you can provide a proper systemic immunity.
Yes. Sir, if that virus is targeting the reproductive system, is it going to definitely target the respiratory system also, or there is no rule like that?
Primary entry is through the respiratory tract. Primary entry will be the respiratory tract. Then the site of predilections may be the oviduct or the kidneys, depends on the virus. Majority of the viruses can affect the oviduct.
Yes, sir. You said at some stage it is not possible to recover the batch to the normal phase.
Definitely, some birds can have a reversible effect. But some birds definitely will have a irreversible effect when the ovaries are affected.
So, at what phase we can take the decision of batch culling, sir? In case of IB, suppose the case is repeating.
That's what when the flock is not going beyond your economical production values, then definitely you can think of the culling. So
even if it is a phase
some of the breeders, the peak production didn't reach 50, 55 or 60. So such definitely flocks are not economically viable to be kept. So some
even
till
if
the market is good, they will be keeping. Pardon?
Even if it is a phase 2 production also.
Yeah, phase 2 production also. And when the flocks are down to a certain extent and they are not recovering back, irreversible damage has happened, definitely.
Thank you, sir.
Thank you, sir. I think many of the questions have been answered in the chat box. Yes. So if there are no questions, I can now proceed.
Yeah, please carry on.
Yeah. Thank you very much, sir, for answering the questions patiently. Now I request Dr. D.K. Day, sir, Vice President of this Association of Avian Health Professionals, to share his concluding remarks.
I think both the subjects were so vast, difficult for me to conclude, because I think both the issues are, kind of a mega issues. And Dr. Prakash Reddy has taken a basically panorama of IB, and he took COVID as well as, IB, and taken the global perspective in India and how challenging is the control program. Yes, there are pioneer vaccine companies in India as well as outside India who are working for a homologous vaccine, which is, according to him, if I've understood, is the right solution. However, vaccine development takes time. And don't worry, I can confirm from the vaccine industry that people are working on it. Solution may not be immediate, but certainly we're working. But as you said, we say IB gets complicated due to the presence of mycoplasma LPAI, and differential diagnosis is also a challenge. So is a control program, but we have to live with it. Same in case of coccidiosis. Dr. Kurkure has given a new perspective that how in depth he has worked upon the different control measures. And yes, when he started presentation, I was also thinking about the litter management, what about water management, and India is a country with humidity, seasonal, so oocyst counts vary from place to place. And yeah, globally, people are shifting from antibiotic to non-antibiotic therapy, vaccines. So I believe, I don't say that we are running against time, but I think we have to run ahead of its time. And we debate more, we discuss more, and assure you that, the pharmaceutical and the vaccine industry, along with the health, avian health professionals, working towards a better solution which is sustainable. Thank you so much. Thank you, both the speakers. Thank you.
Thank you very much, sir. As we came to the end of this session, I now request Dr. M. R. Reddy, sir, Secretary of this Association of Avian Health Professionals, to offer his vote of thanks.
Thank you, Dr. Swati. Good afternoon, everybody. On behalf of Avian Health Professionals, I take this opportunity to propose vote of thanks. At the outset, I thank both the speakers for their excellent presentation, Dr. Kurkure sir and also Dr. Prakash Reddy. And really, their talk is enlightened us. And thank you very much, sir. Then I thank Dr. D.K. Day, Vice President of AAHP, for chairing today's event and for his enthusiastic support. I extend my sincere thanks to Dr. R.N. Chatterjee, Director DPR, for extending the facilities for conducting this webinar. Special thanks to and heartfelt thanks to all the participants, those who have participated actively in the webinar and making the webinar successful. I thank Dr. Swati for moderating this today's webinar. With short notice, she has done the event. Then finally, I thank our IT team, Dr. Prince and his team workers, for arranging the Zoom and also recording the YouTube in the YouTube channel. And this is actually available in the YouTube channel, and I will share the link if anybody wants. With these words, we move to the end of today's webinar, and with one final note, that we have this conference, AAHP Poultry Health Conference, in the February 23rd and 24th next year. So I request and invite all the AAHP life members and also industry professionals to take part in the conference and make it successful. And I thank once again each and every